Students, welcome back to the biology class. In our previous class, we started with the chapter tissues, in which we learned that tissues are of two types, plant tissue and animal tissue. We started off with plant tissues. Plant tissues are further of two types, meristematic tissues and permanent tissues. Meristematic tissues, on the basis of location, they are further of three types, apical meristem, intercalary meristem and lateral meristem. Today we are going to start with permanent tissues. Permanent tissues are formed from meristematic tissues. They do not divide and have permanent shape and size. The process by which cells arise from a meristematic tissue and take up a permanent shape, size and function is called differentiation. So, the process by which a meristematic tissue changes into permanent tissue is known as differentiation. Permanent tissues are of two types, simple tissue and complex tissue. A simple permanent tissue is the tissue which is made up of similar kind of permanent cells that carry out the same function. Complex permanent tissues are made up of more than one type of cells and they work together as a unit. Simple tissues are further of two types, dermal tissue and ground tissue. Dermal tissue. These tissues generally form the outer layer of the plant parts such as stem, roots, leaves, fruits, flowers, etc. It tends to provide protection against adverse environmental conditions and foreign invaders. Dermal tissues are also referred to as protective tissues. The exchange of gases through stomata is also one of the tasks achieved by this tissue. Dermal tissue are of two types, epidermis and cork. Epidermis and cork are referred to as protective tissues. You can see over here epidermis forming the outer layer of the plant body. Similarly, cork. So, epidermis and cork both are referred to as protective tissues. Ground tissue. Ground tissue form bulk of the plant body. It provides support, strength and flexibility to the plants. They are also known as supporting plant tissues. They are of three types, parenchyma, collenchyma, sclerenchyma. We will study each one by one. Parenchyma. Let us first study the nature and structure of parenchyma. Cells present in parenchyma are generally large, oval or spherical in shape. Cell walls are very thin. Cells are not tightly packed. Intercellular spaces are present between the cells. Intercellular spaces are shown over here. You can see some space is there between the cells. They have large central vacuoles. Large central vacuoles are also present in the cells of parenchyma. Location. It is widely distributed in plant bodies such as stem, leaves, flowers and fruits. It is generally located in the soft regions of the plant. It is also found in the packing tissue in the xylem and phloem. Xylem and phloem are complex permanent tissue which we'll be studying in next lecture. Functions of parenchyma. It acts as a packing tissue. It helps in maintaining shape of the plant. Parenchyma found in leaves and green stems help in the process of photosynthesis as they have chloroplasts in them. It also stores food. Transport of material takes place through parenchyma cells. Intercellular spaces in parenchyma also allow gaseous exchange. Collenchyma. Nature and structure. Collenchyma is composed of living cells. Collenchyma is characterized by deposition of pectin and extracellulose at the corner of the cells. So, corner of the cells in collenchyma, they are thickened by the deposition of pectin and extracellulose. Intercellular spaces are generally absent. They are elongated cells. Chloroplast is present in the cells, so they are also able to perform photosynthesis. You can see the structure of collenchyma over here, and these wall thickenings are due to the deposition of extracellulose and pectin. Location. Collenchyma is distributed below the epidermis of dicot stem and petiole. It is found in the midrib of dicot leaves. Collenchyma is absent in monocot stem, root and leaves. Functions of collenchyma. It provides mechanical strength and elasticity to the plant. It gives tensile strength to the parts of the plants where it is located. Students, tensile strength refer to the strength of the material or the stress it can bear without tearing apart. It makes the bending in the various parts of the plant easier without breaking them. The presence of chloroplast helps them to manufacture sugar and starch. Sclerenchyma. Nature and structure. Cells in sclerenchyma tissue are dead and have no protoplasm. A substance called lignin is present in the cell walls of sclerenchyma. Due to this lignin, its walls are very thick. There are no intercellular spaces present between the cells, hence the cells are very tightly packed. You can see sclerenchyma over here. The sclerenchyma cells, there are two types, fibers and sclerites. Fibers are elongated with pointed ends. For example, fibers present in cotton and jute fibers. Sclerites are small, oval or irregular in shape. They have thick lignified walls. For example, they are found in seed coat and pulp of fruits. Location. Sclerenchyma, they are widely distributed in stems, roots, veins of leaves and hard coverings of seeds and nuts. You can just take an example of walnut. Walnut is very hard due to the presence of sclerenchyma tissue in it. Functions. It provides protection and mechanical support to the plants. It helps the plant to withstand various strains as it provides elasticity to various parts of the plants. So students, today we discussed about permanent tissues, and in whole, if we summarize the chapter done till now, we have done plant tissues which are of further two types, meristematic and permanent tissue. Meristematic tissues, they were of three types, apical meristem, intercalary meristem and lateral meristem. So today we have done permanent tissue which are further of two types, simple tissue and complex tissue. Simple tissue, they are further divided under two categories, dermal tissue and ground tissue. Dermal tissue, they are of two types, epidermis and cork. Ground tissue, they are further of three types, parenchyma, collenchyma and sclerenchyma. So students, I hope you have enjoyed today's lecture and you understood it thoroughly. So we'll be meeting in an interaction session tomorrow in which you'll be able to clear your doubts. Till then, stay safe. Thank you.